Welcome friends to a new session on genetics. In this session, we will discuss about extensions to Mendelian genetics and explore laws of inheritance. Let us see about gene interactions. At the beginning of the 20th century, geneticists had no idea about how genes influence particular phenotype. They knew nothing about the structure and function of genes or any techniques to study it. They proposed everything about the action of genes by analysis of phenotypes only. These analyses showed that a particular gene can influence different traits. Some of the first evidence that a trait can be influenced by more than one gene was obtained by William Bateson, who was famous for the study of heredity and biological inheritance, and also for Gregor Mendel's ideas following the rediscovery in 1900 by Hugo de Vries and Carl Correns, as well as Reginald Punnett, who is the creator of the Punnett square from breeding experiments with the chickens. The work of Bateson and Punnett clearly demonstrated that two independent assorting genes can control a single trait. That means, different combinations of alleles from the two genes resulted in different phenotypes. Genes have often multiple alleles. A gene interaction is the interaction of genes which are alleles or non-alleles on a specific phenotypic character. Modified hybrid ratios are the outcome of these type of gene interactions. There are two types of genetic interactions: intragenic, that is interallelic, and intergenic, that is non-allelic. In case of intragenic interaction, two alleles which are located on the same gene locus on two homologous chromosomes interact to produce specific phenotypic expression. A new phenotype was produced in intergenic interaction by the interaction of two or more independent genes present on the same chromosome or different chromosome. Interaction of genes determines a particular biological property. Incomplete dominance and codominance are called intra-allele interaction. Incomplete genetic dominance is similar to but different from codominance. The phenotypic characters of both alleles are expressed completely and produce a new phenotype in codominance. In this session, we consider some of the early findings of Mendel's gene interactions experiments. One of the important findings of Mendel's experiments was that genes can exist in alternate forms. He identified two different allelic states, one dominant and one recessive. Sometimes one allele did everything to determine the phenotype, but the other did nothing. Sometimes these two alleles can affect the same single characteristic. Based on these genetic interactions, Mendel studied 7 traits separately. One is seed color, next seed texture, plant height, flower color, flower position, pod shape and pod color. According to his findings, these genetic interactions have a different effect on the phenotype. In short, Mendel's experiments established that an individual's genotype is made up of many alleles. If the two alleles of an inherited pair are the same, then it is called homologous, or if it differs, it is heterozygous condition. The one which determines individual's specific phenotypic characteristics is called the dominant allele. The other one has no effect on organism's appearance at all. Sometimes the dominant allele hides the phenotypic effect of recessive allele. According to Mendel, the law of dominance stated that the recessive alleles will always be masked by the dominant allele. Therefore, a homozygous dominant plant, that is capital TT, which represents the tall, is crossed with homozygous recessive small t, which represents the dwarf plant. The dominant phenotype will always express as capital T, small t, which is the heterozygous tall one. That means the tall character is controlled by a pair of different characters, that is capital T and the small t. It is important to note that the law of dominance is significant but not universally applicable, because there are many examples where complete dominance is absent. In such cases, the phenotype of the F1 generation is an intermediate between the effects of two genes of those parents' phenotypes. This is seen as an exception to Mendel's law of dominance. Genetics has identified innumerable examples for incomplete dominance in plants and animals. Example, flower color in 4 o'clock plant, which is called as Mirabilis jalapa, and the color of Andalusian fowl. Let us first see about the incomplete dominance in Mirabilis jalapa. According to Mendel's inheritance, a dominant or a recessive allele of single genes controls such traits is a simple hereditary process. However, the dominance of one allele over the other allele is not complete. Incomplete dominance, always known as partial dominance, identifies such circumstances. Incomplete dominance is a form of intermediate inheritance in which one allele for a specific trait is partially expressed over its paired allele. The result is a third phenotype in which the expressed phenotypic character is a combination of the dominant and recessive phenotypes. Incomplete dominance is also known as partial dominance or semi-dominance. Flower petal color in 4 o'clock plant provides a very good example of incomplete dominance. 4 o'clock plant is the most commonly grown ornamental plant, which is the native of America. The 4 o'clock plant gets its common name from the fact that the flower opens up in the late afternoon. When we examine the Mirabilis jalapa, there are 3 colors of flowers. One is red, pink and white. This is the result of interaction among the genes. These interactions include incomplete dominance. Here, a red represented by capital R flowered and white represented by small r flowered varieties were crossed, and the F1 progenies all produced pink colors. The genotype is capital R, small r. The F1 progeny is the intermediate between the two homozygous parents, that is red and white. This type of inheritance pattern is known as incomplete dominance. When the F1 pink flowered varieties were self-pollinated, they produced red, pink and white flowered plants, and the segregation are in the ratio 1:2:1 in the F2 generation. In this case, red color producing gene is not completely dominant over white color producing gene. The effect of the two alleles is more visible. The phenotype is a fine mixture of the two alleles. The expressed hybrid is new, but it has no allele of its own. A quantitative effect is present in incomplete dominance. Geneticists can tell that in this case, flowering time of Mirabilis jalapa is controlled by the interaction of two genes in a single locus, because self-sterilization of capital R, small r plant produces a 1:2:1 ratio in the progenies. Now, let us go to the case of codominance, which we can very well represent with a coat color in cattle. There is another pattern of inheritance, which is also an example of lack of dominance or more than one pattern of dominance occurring under certain circumstances. Codominance is a relationship between two versions of a gene. Individuals receive one version of a gene called an allele from each parent. If the alleles are different, the dominant allele usually will be expressed, while the effect of the other allele called recessive is masked. Codominance implies that there is an independence of allele function. Roan is a pattern common in shorthorn cattle and Belgian blue cattle. In Roan cattle, coat color is inherited through incomplete dominance. Roan is a coat color found in cattle generally defined as a mixture of red pigmented and white hairs. This is a good example of codominance. In codominance, the two alleles are fully expressed or segregated at the same time as in heterozygote in the phenotype. In codominance, two organisms with different phenotypes were crossed. It produced new progenies with a new phenotype having both of the parental traits, that is, both dominant and recessive appear together. The R allele is responsible for the production of red pigment in the coat hairs, while R prime leads to no pigment production. There are alleles for red and white hair in Roan cattle. In animals having the genotype R are red hairs. Animals with R prime genotype appears as white hairs. Animals with heterozygous genotype R primed have some red pigmented hairs and some white hairs and appear as Roan. When a red cattle is mated with white cattle, the cows have a mixture of red pigmented as well as white hairs, giving them an overall color called Roan. These Roan cows are different in color from both the parents. A cross between R primed with R primed phenotype, the cattle produces offsprings that are in the phenotypic ratio 1 red, 2 Roan and 1 white. In this case, both the alleles from parents are more visible. Both the two alleles produced their effect independently without mixing their effect. That means, the expressed phenotype Roan is a combination of the two phenotypes red and white and their alleles. That means, a quantitative effect is absent over here. Now, let us examine the case of lethal genes, which can be very well represented with the example of albinism and coat color in mice. Genes may affect viability as well as the visible traits of an organism. Sometimes organisms die when these alleles or genes are present in homozygous condition. These are called lethal alleles. The phenomenon of activity of lethal genes is called lethality, that is death. Lethal alleles can be either dominant or recessive and autosomal or sex linked. The gene involved is considered as an essential gene. Individuals carrying the dominant lethal allele will die for sure. Lethal effect of an allele may be dominant or recessive. If lethality due to homozygosity for a recessive allele, it is called recessive lethality. In this case, the phenotypic ratio is converted from 3:1 to 2:1. The lethal effect occurring in heterozygous individuals is called dominant lethality. Geneticists believed that mutation can alter only the appearance of an organism after Mendel's law were rediscovered. But it was discovered that a mutant allele can cause the death of an organism. When a specific allele or gene is mutated, it results a lethal phenotype. Lucien Cugnot is well known for multiple allelism at a genetic locus, and he is a French biologist who discovered lethal genes while studying the inheritance of coat color gene in mice. Lucien Cugnot discovered an incompletely dominant allele capital Y for yellow coat color in mice, which is fatal in homozygous dominant condition. When he crossed a yellow mice represented with capital Y with a yellow mice capital Y and expected the progenies in 3 yellow which is represented by capital Y, 1 white small y, but the observed ratio was 2 yellow and 1 white. Then he determined by this experiment that all the yellow mice were heterozygotes, that is capital Y, small y, and it was dominant over the white coat color phenotypic trait. However, no homozygous yellow mice were obtained. These premature dead forms are dominant homozygous. From a genetic perspective, this means that the yellow coat colors are heterozygous and the capital Y genotype is lethal in embryonic development. In 1910, William Ernst Cassel, who was an early American genetist, and Clarence Cook Little, an American genetics, cancer and tobacco researcher, as well as an academic administrator, established Cugnot's discovery of lethal genes by proving that a quarter of the offsprings from the crosses between the heterozygotes died due to the failure of implantation in the uterine lining during embryonic development. 1/4 of the offsprings, that is homozygous yellow mice, died, but Cugnot did not see in his tests. Studies of coat color in mice reveals how genes incorporate to determine the genotypic and phenotypic characters. According to Cugnot's findings, the gene capital Y has multiple effects. The gene capital Y controls the coat color of mice and affects individual viability. That means, the gene capital Y is dominant over coat color and recessive with respect to lethality, but the heterozygous yellow has genotype capital Y small y, which somehow managed to survive. Albinism is an example for lethal factor in mice. 5 different types of genes interact to determine the coat color in mice, that is A, B, C, D and S. The gene A determines the distribution of pigment in hairs. The wild type allele capital A produces a phenotype called agouti, an overall greyish color or salt and pepper appearance. This is due to the band of yellow color on the otherwise dark hair shaft. The allele small a determines the non-agouti phenotype. The yellow band is absent, there is solid dark pigment throughout. The capital B gene determines the color of the pigment. The 2 major alleles codes for the color of the pigment. Capital B codes for the black pigment and small b for brown. The allele capital B gives the normal agouti color in combination with A allele, but gives solid black with small a. The capital A and small b genotype gives a striped brown color called cinnamon, and the genotype small a, small b gives solid brown. Now, the inheritance pattern of the A and B genes are as follows. when cinnamon is crossed with the black, or if agouti is crossed with brown, in the F1 generation you get agouti. And if agouti and agouti is crossed together, in the F2 generation you get the progenies in the ratio 9:3:3:1. 9 is agouti, 3 is cinnamon, 3 is for black, and 1 is for brown. The capital C gene, wild type, is responsible for the color expression, and the small c gene prevents color expression. The small c genotype is epistatic to the other color genes lacking coat pigment are called albinos. Another allele of the C gene is the C superscript H, which is the Himalayan type. The C superscript H allele is the heat sensitive version of the small c allele. The CH is functional only at the colder body extremities and makes color pigment. It behaves just like the albino allele small c in warm parts of the body. The small c constitution produces a standard recessive epistasis modified ratio in the following cross. Now, we look into a cross between albino and brown or black with albino. In the F1 generation, the progenies are black. Now, when the black and black are crossed together, in the F2 generation, we get a progeny ratio of 9:3:3:1, wherein 9 is for black, 3 is for brown, and 3 again for albino and 1 for albino again. Genetic analysis of coat color in mice reveals that the distribution of pigment to the hair is due to the activity or interaction of many genes and where they are displayed as coat color. Different interactions between these genes can modify the color expression. Here we focus just one gene, the C gene, capital C gene, responsible for coat color in mice. The capital C gene produces the enzyme tyrosinase. This enzyme is active in the pigment synthesizing biochemical pathway which gives coat color in mice. Albino mice occur due to a recessive mutation of the capital C gene. The albino phenotype masks any phenotype that might be caused by the recessive homozygous small c. The capital D gene determines the intensity of the pigment of the hair. The genotypes capital D and capital D small d permit the complete expression of coat color in mice, but small d genotype dilutes the color and looks like milky. This is due to an uneven distribution of pigment in the hair. The possible coat colors are dilute agouti, dilute cinnamon, dilute brown, and dilute black. This is another example of a modifier gene. Here the dilute black is crossed with brown or black is crossed with dilute brown, and the F1 progenies are black. When the black progenies are crossed between each other, we get F2 progenies in the ratio 9:3:3:1, the 9 representing the black, 3 representing dilute black, 3 representing brown, and 1 representing for dilute brown. The capital S gene controls the distribution of pigmentation, that is, the presence or absence of spots throughout the body. The genotype capital S results in no spots, whereas small s is responsible for a spotting pattern called piebald in mice. Let us summarize the foregoing discussions of gene interactions. The chromosome contains the genes that inherit from parents to offsprings. There may be different forms of the same gene called alleles. These genes determines the phenotypes of the offsprings, which depend on several factors, including what genes they inherit and whether they are in dominant or recessive condition. Mendel derived many inheritance patterns. An individual have an intermediate phenotype of both parents, and the inheritance pattern is called incomplete dominance. Mirabilis jalapa is an excellent example for incomplete dominance. In incomplete dominance effect, the two alleles is more conspicuous and produces a fine mixture of two alleles. Expressed phenotype is new. It has no allele of its own and has quantitative effect. In a heterozygote, both the alleles of gene are fully expressed and they are called codominance. Coat color in Roan cattle is the best example of codominance. In this effect of both the alleles are clearly visible. The two alleles produce their effect independently without mixing. The expressed phenotype is a combination of the two phenotypes and their alleles and quantitative effect is absent. Sometimes the alleles or genes present in homozygous condition cause an organism to die. These genes are called lethal genes. Cunard studied the genetic basis of yellow coat color in mice, which is a good example of lethality. In wild mice, the normal coat color is produced by a complex set of genes which interact and determines the type of pigment, its distribution in the individual hairs, its distribution pattern on mice and the absence or presence of pigment. These types of interactions that modify the normal coat color in mice and the pigment distribution pattern. These interacting genes in mice determine the most characters in any organism. Different kinds of modified dihybrid ratios point out how these genes can interact with each other to determine phenotype of an organism. Now, let me give you some assignments to work out. 1. Explain how genes and their interactions produce a range of different phenotypes. The second one is write down different kinds of modified dihybrid ratios. Coming to the third, what are the factors which causes variation in phenotypes? A fourth one would be explain how the coat color in cattle follows a codominant inheritance pattern. The fifth one is find out some important applications of gene interactions. And the last assignment would be explain the gene interaction in coat color in mice. Now, I can also suggest you some books for your further reading. First one is entitled Mendelein Ratios and Lethal Genes, written by Lobo and published by Nature Education in the year 2008. Second book is entitled Principles of Genetics, written by Gardner and Snustad in the year 1984, published by John Wiley, New York. Third book is entitled Cell and Molecular Biology, written by D. Robertis and Robertis in the year 1991 and published by Leah and Febiger, Washington. Fourth book is entitled Genes, written by Levin in the year 1994 and published by Oxford University Press, London. Fifth reference book would be entitled Chromosome Technique Theory and Practice, written by Sharma and Sharma in the year 1980 and published by Aditya Books, New Delhi. Yet another book would be entitled Principles of Genetics, written by Sinnott, Dunn and Dobzhansky. It is published by McGraw-Hill in New York. Now, let us look into some websites also where you can get information related to these aspects. One is www.genetics.org, theagricos.com. We have ended up this session, and thanks for being with us. We will meet again in another session. Till then, bye.